So imagine you were standing in a neonatal intensive care unit.
Right.
You're looking at this tiny newborn, but this infant is, well, the one in 3,500. They're born with a congenital defect called esophageal atresia.
Yeah. It's a truly heartbreaking scenario for any family to face.
It really is.
Yeah.
And specifically, we're talking about long gap esophageal atresia, or LGEA. A massive section of their esophagus simply doesn't exist.
Yeah.
There is just, no connection to the stomach.
None at all.
They can't swallow. They can't eat. I mean, even their own saliva is this constant, terrifying aspiration risk.
And, it is one of the most mechanically demanding challenges in pediatric surgery today because the current treatment algorithms we rely on, they're brutal.
Yeah, we aren't just talking about, slipping in a temporary feeding tube and waiting.
No, not at all. We're talking about intensive, highly invasive procedures like the Fokker process.
Oh, right, the traction one.
Exactly, where we surgically place internal traction sutures and basically mechanically stretch the two blind ends of the esophagus over weeks, just agonizingly pulling them millimeter by millimeter until they can finally be stitched together.
Which just sounds so traumatic for a tiny infant.
It is. Or, another option is we perform a gastric pull-up. We physically relocate the infant's stomach out of the abdomen and pull it up into the chest cavity.
Wow.
Yeah. The morbidity is staggering, the complication rates are sky high, and, the quality of life for these patients is fundamentally compromised for years.
But what if, and this is why we're here today, what if you could just grow a replacement? Not like a rigid synthetic plastic pipe, but a dynamic, living, innervated tube that actively swallows.
That is exactly the focus of our deep dive today.
Right. We are unpacking a landmark 2026 paper from Nature Biotechnology. These researchers, they successfully engineered a 2.5 centimeter functional esophagus.
And they achieved what has essentially been the holy grail of tissue engineering here.
Independent peristalsis and muscle contraction. Plus, and this is huge, without lifelong stents, and crucially, without any immunosuppression.
Completely avoiding the need for anti-rejection drugs.
Exactly. We are going to completely deconstruct the exact cellular mechanisms, the transcriptomic shifts, and the messy surgical algorithms that made this medical marvel possible. So, okay, let's unpack this. If we're building an organ, we obviously don't start with just thin air. We need a scaffold.
Precisely. You can't just throw cells into a void. You need a highly organized blueprint that already, understands the complex geometry of an esophagus.
Right, needs to know it's a tube.
Exactly. So the researchers started with a 5 centimeter segment of esophagus, and they harvested it from a 10-kilogram donor pig.
Okay, a pig. But wait, the obvious physiological barrier here is xenotransplantation, right? I mean, you can't just implant raw, living pig tissue into another species.
No, absolutely not. It's actually even more aggressive than a standard human-to-human immune response.
Because of the specific antigens.
Right. Pig cells express the specific carbohydrate antigen on their surface called galactose alpha 113 galactose. We usually just call it alpha-gal.
Alpha-gal, right, like the tick bite allergy thing.
Exactly the same molecule. Humans and, many other primates, we naturally produce incredibly high levels of antibodies against alpha-gal.
So the second it enters the body.
It's immediate. If you introduce that pig antigen, those host antibodies bind almost instantly. It triggers this massive complement cascade.
Which causes hyperacute rejection, right?
Yes. The blood vessels thrombose, they just completely clot off, and the implanted tissue literally turns black and necrotic within minutes.
Within minutes, wow.
So to prevent that, the researchers had to implement a really rigorous 10-day decellularization protocol. They used a modified detergent enzymatic treatment, or DET.
DET. So they're essentially, it's like they're chemical power washing the tissue.
That's a great analogy, but it is far more sophisticated on a molecular level than just a simple wash.
How so?
Well, first, they perfuse the tissue with a 4% solution of sodium deoxycholate for 48 hours.
And that's the detergent part.
Correct. It's a really powerful anionic detergent. On a molecular level, it physically inserts itself right into the lipid bilayers of the cell membranes.
Like soap cutting through grease.
Exactly. It absolutely solubilizes them. It tears the pig cells completely apart, but destroying the membranes isn't enough because you still have the internal cellular machinery left over.
And most dangerously, the chromatin, the DNA, right?
Right, the genetic material.
Because, leftover fragmented DNA can still be highly immunogenic. It can act as a, what is it called, a damage-associated molecular pattern? A damp.
Yes, exactly, a damp.
Which basically sets off massive inflammatory alarms in the host body.
Which is exactly why the second step utilizes an enzyme called DNase.
Okay, so detergent first, then enzyme.
Right. The DNase acts like molecular scissors. It gets in there and cleaves the phosphodiester bonds, basically chewing up all that residual genetic material into harmless pieces.
So they run multiple cycles of this detergent-enzyme combo.
Over the course of 10 days. And then it's followed by a final sterilization phase using gamma irradiation.
Wait, why gamma irradiation?
Hmm?
Why not just use something standard, like chemical sterilization or just extreme heat?
Well, heat would completely denature the structural proteins. It would melt the physical architecture they're trying to save.
Oh, sure.
And chemical sterilants, like ethylene oxide, they can leave behind these toxic micro-residues.
Which would kill the new cells you want to put in later.
Exactly. So gamma irradiation is perfect because it deeply penetrates the tissue, ensuring total sterility without altering the mechanical properties of what remains.
And what remains is the critical part. It's pure extracellular matrix, or ECM.
The ECM, yes.
So, okay, if I'm tracking this molecular deconstruction correctly, we've stripped away the lipid bilayers, we've chewed up the DNA with molecular scissors, and we've totally eradicated those alpha-gal antigens.
We have.
So we're left with this invisible stealth organ frame. It's a matrix composed entirely of structural proteins like collagen, elastin, and complex glycosaminoglycans.
Right.
So it's basically like taking a fully furnished older house, ripping out the drywall, ripping out the wiring, the plumbing, and, totally evicting all the residents until literally all you have left is the pristine, non-immunogenic wooden framing.
That is the perfect way to visualize it.
And the host body just sees it as structural scaffolding, not as a foreign invader at all.
Exactly. The decellularized ECM is a completely blank biological canvas.
Okay, so let's do a quick recap for you listening just to lock in this first critical phase. Here are the bullets. 1, we take a donor esophagus from a pig. 2, we use a 10-day DET protocol, that detergent and enzyme combo, to destroy every single cell and piece of DNA. And 3, we keep that extracellular matrix scaffold perfectly intact. So, listener, here is a question for you to chew on. Think about a building being stripped completely down to its studs. What kind of tenants would you want to move into this new structure to ensure it actually functions as a living, moving house again?
And that leads us perfectly into the next phase, because a structural canvas is useless on its own.
Right.
If you implant just that bare scaffold into a chest, it will eventually just fibrose, scar over, and collapse. It requires a living, active workforce to maintain it.
Which brings us to the cellular paint, the new tenants. And the researchers, they didn't just grab, generic stem cells off a shelf.
No. They were incredibly specific. They took a very deliberate 5x5 millimeter biopsy.
It's a tiny square.
Right. From the rectus abdominis, the abdominal muscle, and the fascial tissue surrounding it, they isolated 2 distinct cell populations from that biopsy and mixed them in a really precise 7:3 ratio.
7 parts mesoangioblasts to 3 parts fibroblasts.
Exactly. MAs and FBs.
Let's unpack those 2 groups. So the fibroblasts, the FBs, derived from that fascial layer, they act as the resident maintenance crew, right?
Yes. Their biological imperative is to constantly secrete and organize new extracellular matrix. They continually repair and remodel that wooden house frame we just talked about.
But the mesoangioblasts, the MAs, those are the true driving force of this whole organ regeneration.
They are the stars of the show. MABs are parasite-like, bipotent myogenic precursors.
Okay, let's slow down and unpack that lineage for a second. They are parasite-like, meaning their natural niche in the body is usually wrapping around the endothelial cells of microscopic blood vessels, right?
That's where they normally hang out, yes.
But they are bipotent myogenic precursors, meaning they have this inherent plasticity to differentiate down 2 very distinct muscular paths.
Yes. Depending entirely on the microenvironmental cues they receive, MAs can become either smooth muscle,
which provides that involuntary, sustained slow contraction necessary for lower esophageal peristalsis.
Right. Or they can become skeletal muscle, which drives the rapid voluntary swallowing mechanism up in the upper esophagus.
That is so clever. So to populate the pig scaffold with these cells, the team meticulously executed exactly 120 separate microinjections.
Spaced out incredibly precisely.
Yeah, exactly 3 millimeters apart along the entire tube, delivering 10 to the 5th cells per microliter.
The exact spatial precision there is vital, because if you just lazily dump the cells into the center of the tube, they won't penetrate that dense collagen matrix.
They just wash away.
Right. And if you inject them too densely in one single spot, they compete for nutrients, they starve, and they undergo necrosis.
But here is where the methodology gets really intense.
Yep.
They don't just meticulously seed the scaffold and then immediately drop it into a patient.
No, that would be a disaster.
Right. They subject the engineered graft to a full 1-week incubation inside a dynamic flow bioreactor.
And we have to emphasize, the bioreactor is not merely a holding tank. It's not just a petri dish. It is a critical conditioning environment.
What's that?
It physically spins the graft, like on a rotisserie, while simultaneously pumping fluid dynamically through the inner lumen and over the outer surface.
So it's simulating the real body.
Exactly. This introduces mechanical shear stress, which triggers these massive mechanotransduction pathways inside the newly injected cells.
And the researchers actually proved this mechanical conditioning works. They used single-nucleus RNA sequencing, or snRNA-seq, to map the entire transcriptomic shift before and after the bioreactor phase.
They looked at the actual gene expression.
But, snRNA-seq generates this massive wall of transcriptomic noise. It's literally millions of data points. When you actually look at that data, what specific pathways prove this spinning liquid bioreactor did anything useful?
It's fascinating. The entire transcriptomic landscape completely reorganized itself to favor a pro-angiogenic phenotype.
Angiogenesis being the creation of new blood vessels.
Yes. After 7 days in the bioreactor, the expression of key genes, specifically VEGF-A, HIF-1A, PTGS2, and NDRG1, they absolutely surged.
They went through the roof.
Right. And HIF-1A is particularly important here. It stands for hypoxia-inducible factor 1 alpha. You see, the bioreactor fluid environment is naturally lower in oxygen than the native bloodstream would be.
Oh, okay.
This mild hypoxia actually blocks the degradation of the HIF-1A protein, stabilizing it inside the cells.
Which essentially forces the cells to shift their entire metabolism. They downregulate oxidative phosphorylation, which needs a lot of oxygen, and they massively upregulate glycolysis.
Exactly. Burning glucose without relying on oxygen.
It is essentially astronaut training in a simulator.
I love that analogy.
Right. The bioreactor intentionally starves these cells of oxygen just enough to trigger their ultimate survival pathway. It teaches them to scream out chemical signals for new blood vessels the very second they eventually get implanted into the dark, oxygen-poor cavity of the chest.
They are preconditioning them for the physiological shock of surgery.
That is wild.
It is. That metabolic shift is literally the difference between a graft surviving and a graft just dying of ischemic necrosis on day 1. By the time that tube leaves the bioreactor, those cells are biologically primed and ready to recruit a massive host vascular supply.
Okay, wow. Let's map out exactly where we are right now before we head into the operating room. Here's your 10-minute recap. 1, we harvested the patient's own autologous MEs and FBs. 2, we precisely microinjected them into the pig scaffold. 3, we trained them in a spinning bioreactor to survive low oxygen and promote blood vessel growth.
A perfect summary.
So listener, put yourself in the shoes of the thoracic surgeon. You are handed this delicate, newly seeded, laboratory-grown tube. How do you protect it the exact moment you slice open a breathing, moving chest cavity and stitch it in?
And that transition from the highly controlled environment of a bioreactor into the chaotic, messy physiological reality of an in vivo model, that is where the vast majority of tissue engineering simply fails.
It just falls apart in the real world.
Usually, yes. To test this realistically, they utilized 10-kilogram Göttingen mini pigs. It's a widely accepted translational model because their esophageal dimensions and, crucially, their intrathoracic pressures closely mimic human pediatric patients.
And to address that immediate need for blood supply that you just mentioned, the one the cells were screaming for, the surgeons utilized this classic, highly effective technique called the pleural wrap.
Yes. They took a flap of the pleura, which is the highly vascularized membrane that natively surrounds the lungs,
Right.
and they physically wrapped it a full 360 degrees around the engineered graft during implantation.
So they basically swaddled it.
They did. That pleural wrap acts as an immediate biological blanket, just rich with capillary networks. It provides the crucial initial hemodynamics required to sustain those oxygen-starved cells until true intrinsic angiogenesis can fully take over.
But even with the pleural wrap providing blood, a freshly seeded ECM tube is still structurally super vulnerable. I mean, without the negative intrathoracic pressure of a fully mature, thick muscular wall, the graft would simply collapse on itself every time the pig took a breath.
It would act like a wet paper straw.
Right. So to fix that, they deployed biodegradable transanastomotic PDS stents. basically physical plastic tubes placed inside the lumen of the esophagus to physically hold it open while the surrounding tissue integrated.
Yes, stents are a necessary evil here.
And here is the part of the paper I actually appreciated the most.
Oh, the complication reporting.
Yes, they did not hide the complications. They didn't pretend this was some flawless, magical science fiction procedure. The clinical reality was incredibly messy.
It was heavily complicated, absolutely, but it was highly informative. Looking at the clinical data, they did achieve 100% 30-day survival.
Which is amazing.
It is. However, only 63% so. 5 out of the 8 pigs actually reached the full 6-month endpoint of the study. The postoperative challenges they faced mirrored what we see in human pediatric intensive care units almost exactly.
Like what?
Well, for instance, the pigs engaged in pica. They started eating their own bedding material.
Oh, wow.
Yeah, which caused severe foreign body obstructions right inside the healing graft.
But even beyond the behavioral issues of the animal model, the internal PDS stents themselves caused significant pathology. They triggered hyperplastic epithelial polyps.
Yes, and this is a classic physiological response to mechanical friction. The rigid plastic stent constantly rubs against the newly regenerating inner lining of the esophagus.
It's constantly irritating it every time they move.
Exactly. This sheer stress signals the basal epithelial cells to hyperproliferate. They rapidly multiply to form these thick, fleshy polyp blockages inside the tube.
But the researchers, they didn't just abandon the grafts when this happened. They treated them using standard clinical algorithms.
Right, they managed it medically.
They administered oral systemic steroids to, reduce the inflammatory cascade that was driving the hyperplasia. Or they literally went in endoscopically with a camera and physically resected the polyps.
Furthermore, when the stents naturally started to migrate and cause strictures, which are severe narrowings of the scar tissue, they performed balloon dilation.
Popping a balloon inside to stretch it open.
Exactly, inflating a catheter to mechanically stretch the lumen back open. And that is the most crucial takeaway from this whole surgical phase of the study.
The complications are fixable.
Yes. The morbidities were significant, yes, but they were entirely manageable using existing standard endoscopic techniques. And most importantly, despite all those strictures and polyps, the surviving minipigs eventually achieved complete stent independence.
That is massive. The tissue fully integrated, the stents degraded or were physically removed, and the animals transitioned to a normal, solid diet. They started gaining weight at the exact same trajectory as healthy, non-surgical control pigs.
It functioned like a normal esophagus.
So, listener, let's do our 15-minute recap. Think about what this means. 1, we implanted the graft and secured blood flow with a pleural wrap. 2, the stents were vital but caused treatable issues like polyps and strictures. 3, the grafts eventually supported normal growth without stents entirely.
It survived the real world.
It did. But here's your next question. An esophagus is not a passive PVC pipe. Just because it survives doesn't mean it functions. How do we definitively prove this engineered tissue isn't just a stiff, lifeless tube of collagen? How do we prove it possesses the actual neurological and muscular architecture to actively dynamically swallow?
Well, answering that requires multimodal evidence. We have to look at histology, spatial transcriptomics, and real-time physiological testing.
Let's start with histology.
Sure. So looking at the tissue architecture under a microscope at the 6-month mark, the wall thickness of the engineered graft had dynamically remodeled itself to perfectly match native esophageal tissue.
It bulked up.
It did. A mature, stratified squamous epithelium had formed all along the inner lumen. That provides a durable, multi-layered barrier against food friction and stomach acid.
And more importantly, using immunohistochemistry, they stained the tissue for a specific protein called beta-3 tubulin, which is a highly specific marker for neural tissue.
And the beta-3 tubulin lit up across the graft.
Yes. Host nerve fibers had actively grown from the pig's body deep into the engineered matrix, providing the electrical wiring necessary for actual movement.
Which is incredible on its own.
But for me, it's the timeline of the muscle regeneration that I found absolutely mind-blowing. When you look at the gene expression data over those 6 months, you see a highly staggered, very specific maturation process.
It doesn't all happen at once.
No. The smooth muscle markers genes like TGLN, DES, and MYH11, they surge early, right around the 1-month post-op mark. But the heavy chain markers for skeletal muscle genes like MYH3 and MYH7, they don't significantly activate until way later, like 3 to 6 months in.
It is a highly distinct temporal sequence.
It perfectly follows the mnemonic. Smooth early, skeletal late.
Smooth, early, skeletal late. I like that.
Right, but the reason that matters so much isn't just because it's a neat timeline. It's because smooth early, skeletal late perfectly recapitulates exactly how the esophagus develops in a human fetus.
Ah.
The bioengineered tissue didn't just grow randomly. It tapped into ancient, highly conserved developmental biology algorithms to basically rebuild itself from scratch inside the pig.
It is profound evidence of the body's innate regenerative capacity once you give it the right cues. However, genomic markers and histological stains, they only tell you what the tissue is. They don't tell you what the tissue actually does. To validate actual contractile function, the researchers had to perform ex vivo organ bath analyses.
Explain the organ bath.
They excised cross-sections of the matured graft, suspended them in a physiological saline solution in the lab, and subjected them to electrical field stimulation. They also use chemical stimulation, applying carbachol, which is a really powerful cholinergic agonist.
And the tissue physically twitched. The engineered muscle generated real, measurable force. It responded to both electrical depolarization and chemical neurotransmitters exactly like native, healthy esophageal tissue would.
It did, but the ultimate, definitive, undeniable proof came in vivo, assessing the intact organ inside the living, breathing animal.
This is the manometry test.
Yes. They utilize high-resolution impedance manometry, or HRM. This involves passing a very specialized sensor-laden catheter down the esophagus of the pig.
And what do the sensors do?
They map both the physical pressure generated by the squeezing of the esophageal walls and the electrical impedance, which perfectly tracks the movement of liquid and gas flowing past the sensors.
So they literally shoot boluses of saline and air into the pig's throat and watch the readouts on the sensors.
And what did they find?
The HRM detected secondary peristaltic waves. The engineered tube was reflexively detecting the mechanical stretch from the fluid and actively sequentially squeezing the bolus downward toward the stomach.
It was generating coordinated propulsive force, not just randomly twitching, but performing a complex sequence.
It swallows.
It swallowed. By sequentially combining a decellularized, non-immunogenic pig matrix, an autologous population of bioreactor primed stem cells, and strategic surgical hemodynamics with that pleural rep, they produced a functional innervated swallowing organ.
And all of this without using a single immunosuppressive drug.
It's a paradigm shift.
So listener, what does this all mean for you and for the broader landscape of medicine? It means the standard of care for severe tissue deficits, whether from congenital birth defects like LGEA or, catastrophic caustic injuries from swallowing chemicals or even massive resections due to cancer, it is fundamentally shifting.
We are rapidly moving away from brute force mechanical management.
Stretching tubes and pulling up stomachs.
Exactly. And we are stepping firmly into an era of true, instructed biological regeneration.
And that leads to one final, deeply provocative question that the paper openly acknowledges, but, cannot entirely resolve.
The tracking issue.
Yes. Because the researchers used autologous cells, meaning cells from the mini pigs themselves, they couldn't permanently tag the injected mesoangioblasts with fluorescent tracking dyes.
Right, because the pig's immune system would eventually recognize the chemical dye as foreign and clear those specific cells out over the 6-month study.
Exactly. Which leaves us with a massive biological mystery. We know for a fact we injected millions of MEBs into that pig scaffold. But did those specific injected cells actually differentiate and physically become the mature muscle fibers that ultimately squeeze the food down? Or did they simply act as temporary construction foremen? Did they just sit in the scaffold for a few weeks, scream out paracrine chemical signals to recruit the body's own host stem cells to migrate in and do the actual building, only to quietly die off once the job was underway?
We actually don't know whose cells built the final house.
We don't.
It challenges our entire conceptual framework of regenerative medicine. Are we literally providing the physical building blocks, or are we merely providing the transient biochemical instructions to wake up the host's innate healing capacity?
It's incredible. So think about that mystery the next time you sit down to eat. As you swallow your food, consider that complex, invisible, autonomic wave of muscle actively carrying it down into your stomach. And think about that one infant in 3,500 sitting in a neonatal ICU right now. Because very soon, the devastating reality of their diagnosis might just be permanently rewritten by a ghost town pig scaffold, a handful of prime stem cells, and the spinning current of a bioreactor. Thanks for taking this deep dive with us. Keep questioning the mechanics of everything around you.